Hello, I wanted to make a little video that would review our exercise in blood vessels and blood pressure in case we can't meet face-to-face, or it can also serve as a pre-lab. Now these are my notes, and so there's text boxes in them, and I will move them to the side when I need to move them to the side. This first text box reminds me that the majority of this lab is spent on introducing a research project. And so it's a fairly short lab of in-class activities because much of the time is spent on talking about our research project. So there's basically three exercises here. We're going to look at artery and vein, we're going to look at atherosclerosis, and we're going to look at major blood vessels in the cadaver or on a model or in some kind of atlas. So first, I'd like you to look at artery versus vein, and it's kind of difficult to interpret this because our microscopes have a kind of a small field of view, and so you can't see everything. So I'll try to fill that in for you. This is an artery right here, and this artery would be extending up and around like this. Down here is the vein, and the vein would be extending down here and around there. What you'd also notice if you looked at this through the microscope is the artery is very circular, and it's very kind of elasticy. And actually there's plenty of elastic fibers in here and collagen fibers. If you remember your three different fibers, collagen is strong, elastic is stretchy, and reticular is kind of like a fishing net and is found in lymph nodes. When looking at the slide, you should be able to notice the three layers. The tunica intima is a very thin layer. It's very, very thin. It's of simple squamous cells, and it's found in both artery and vein, and it's thin in both of them. Its job is to just be smooth. And so it's pretty similar between artery and vein. The next layer goes from here to here, and that's tunica media. That's muscle. And that muscle is used to give strength and also change the size of an artery. It's very thin in a vein because veins don't really change their size. And then this outer layer is called the tunica externa, and that's there for strength. You kind of want to remember that ordering because that stands in almost every other different hollow organ. So the inside does what it needs to do. It's usually smooth, like the inside of the heart is smooth, the inside of blood vessels is smooth, inside of the digestive system is smooth. Then the next layer out provides function. The function in an artery is the muscle. In a heart, it's the muscle. In the digestive system, it's to make pumps so that you can pump in nutrients. And then the next layer out is always strength. So the outer layer of the heart is the strength, the outer layer of the artery is the strength, and the outer layer of the digestive system, whether you're talking about the esophagus, the small intestine, the large intestine, the outer layer provides strength. You'd also notice other things, again, like I said, the artery, if you were to look at the artery on a cadaver, and if you were to squeeze it, it would feel like a latex tube. So it would compress when you squeeze it, but then it would bounce back. Our vein, on the other hand, feels kind of like a thick plastic, like a freezer Ziploc bag. So the veins are very collapsible, and they're very distensible, so they will collapse. They're usually larger too, because about 60 to 65% of your blood is stored in your veins at any given time. Our next exercise is to look at atherosclerotic plaques, and so this is a plaque in the wall of a vessel. Again, a microscope has kind of a narrow field of view, so over here is the lumen, or the lumen could be over here too, but my point is that this here is tunica media, and I'm going to have to go to Wikipedia to find a figure that helps you out with that a little bit more. So first of all, I like this slide because it shows how plaque develops over time. In your 20s, you very well, if you're not taking care of your blood pressure, or if you're eating a high-fat meal and don't have a good diet, and you're not paying attention to HDLs and LDLs and all those kind of things, then you might have plaque starting to build up. And then it builds up and builds up so that in your 50s and 60s, that's when you get atherosclerosis. If you have a chance to go in and see our cadaver, sometimes squeeze an artery, and sometimes you can find these plaques, and they'll literally crackle because not only is there cholesterol in here, but there's calcium in here as well. So this actually calcifies into something that feels like bone, kind of like really thin chicken bone. So when you squeeze one of those plaques in the cadaver, you very well can hear them crack and crackle. So this is an artery. This is the tunica intima. This is tunica media, so muscle. This is tunica media, so muscle. And then this is tunica externa. And what I'd like you to note is that clot forms right in the middle of the tunica media. I don't like that it's a misconception that a lot of people think the plaque just kind of hangs out on the inside of the blood vessel and then it can break loose and that's the clot that causes strokes. That's not true at all. In fact, I'm really frustrated that the Pearson textbook has always used as a metaphor a rusty pipe, that if you have rust on the inside of a pipe, that's what a plaque is like, and that's not true at all. The plaque is not hanging on to the inside of the vessel. It's deep in the tunica media, and the reason I think that's important to recognize is it gives you an idea of the permanence. So not only is this cholesterol, It's also calcium, so it's calcified into almost bone, which means, again, you can't start worrying about this stuff when you get 40 and 50 and your high blood pressure starts to go up. You want to start thinking about this earlier on in your life because it's kind of a permanent process once it develops. It's really hard to get rid of it. Okay, so that's the second thing I'd like you to do, is look at that atherosclerotic plaque. Then the third thing would be to find these arteries and veins and know where they are. Now, probably the best way to do it would be to go to the Prezi. And at the end of the Prezi, notice there's some figures, or you could also find these on Wikipedia because that's where I pulled them from too. So you should be able to find everything on that list here. Ultimately, the way I'm going to test you, though, is I'm going to test you with Anatomy and Physiology Revealed. So I'm in student view here. So you'll go to Anatomy and Physiology Revealed. So this is going to be an atlas of the human body. And for each of those, I'll pick an easy one. I'd want to put in your artery. Might have picked the wrong one. Renal artery. There we go, that's good. So that's the renal artery. This is how I'm going to test you, is use APR. I mean, if we were meeting face-to-face, I'd test you on the cadaver, but if we're not meeting face-to-face, this is how I'll test you. And so normally, anything in bold would be on the cadaver, and anything else could be on a picture or a model. But in this case, obviously, we have to do everything on a model, or we have to do everything on Revealed, so we'll just do it on Revealed. All right, so that's everything for the blood vessel lab.